This program is a presentation of UCTV for educational and non-commercial use only.
This morning we talk a lot about biomarkers, so I feel it would be good to put everything together so that some of the students here in the audience can really appreciate what appear to be disparate different fields can actually come together in more integrated context. So I want to sort of take upon myself to for this task. Now, the way I want to organize my talk is first, get it out there, what are the various definitions of biomarkers and how the entire field, from my perspective as epidemiologist, has evolved over time, and then some of the difficult issues and opportunities in the entire field of molecular epidemiology of complex disease, and discuss some of those related issues within our own research application, and some of the, I would use some of the related works that over the past 10 years that our groups have been involved in to illustrate some of the ideas and concepts. And again, at the end, I would, because of really the mission of this symposium is trying to bring everything together and provide some perspectives for those of you who really, like I said again and again, have the ambition and also abilities to move forward for the next generations of integrated research with emphasis on prevention. Now, even among scientists within this campus, if you talk about biomarkers, it really involves different neurons in your brains. I listed here is several authoritative versions of the definitions of biomarkers. One in 1987 by the National Research Council defined biomarkers as indicator signaling events in biological systems. 15 some years later, by the NIH Working Group, a little bit more formal and systematic way of defining biomarker as objectively measure and evaluate as indicators of normal biological processes, pathologic processes, or pharmacologic responses to a therapeutic intervention. And fast forward for another 4 years, then you come to a new, entirely different set of biomarkers now, as just simply complex imaging modality. Okay, so biomarkers definitions have changed, and I think it would really be helpful if you look at this in a historical context and how he evolved over time, and disease focus and disciplinary perspective. In fact, go back almost an entire century, as early as 1910 or so, when in the study of infectious disease, where we're trying to identify the specific pathogens, either through some simple skin tests, or for example, one example come to mind is the tuberculosis example, to later on some of the nutritional deficiency diseases such as vitamin C or niacin deficiencies, where we can really pinpoint that particular agent as biomarkers, then one can actually identify the etiology for that disease. And not until the 1950s, in fact, when the concepts of multiple risk factors, biomarkers, come to play as a way to study the so-called multifactorials of vascular disease, specifically cardiovascular disease, when it really emerged in mid-centuries, become the mankind's single most important killer. And some of the examples later on, was also alluded to, is the in the cancer field. Now, another point that I wanted to make too is that throughout this morning, you have heard a lot of the sort of concept or discussion about the very fundamental concepts of interaction, gene-environmental interaction. And I think it would be good to actually pay attention to some of these examples at the very fundamental level of some inborn error metabolisms that you have heard about that illustrate the fundamental principles of this interaction between gene and nutrients. In fact, some of the nutrients are really governed by our genetic system, and even in the metabolism process, one single gene defect could cause some of the clinical phenotypes, some of which are very severe, and that is actually created some of the paradigm basis for screening and prevention. But it also helped to illustrate the point that at the most fundamental level is interaction as well as for prevention purpose, and you don't even need to manipulate the particular genotype, but you can actually, by eliminating the specific nutrient or chemical itself, can help cure those diseases. One thing come to mind is PKU. But any examples like this is actually very helpful to illustrate the concept of interaction. Now, again, I take upon myself to, again, illustrate how we have shifted over time from these traditional understandings of the entire disciplinary science of epidemiology to more recently what we call molecular epidemiology, with the idea, again, to study the determinant and distributions in human population, to understanding the pattern of frequency change over time, but ultimately our own design to understand what exactly is in the process that affect that black box that leads to exposure. Like exposure here broadly defined can be gene, can be environment, and can be their interactions that ultimately causes disease. And that's essentially what molecular epidemiology is all about. And then I would also further argue that these markers essentially can broadly also classify for understanding purpose. Has nothing to do with, I mean, obviously some of these categories are not mutually exclusive, but nevertheless help us to identify what exactly are the major markers. And ideally, one wanted to find a kind of biomarkers that not only can serve as indicator for objectively reflecting the expression of particular exposure or interaction, but also markers for susceptibility as well as for disease and prognosis as well. And those are the challenge in identifying those truly important markers, especially given the fact that, now this is the slide that I got from Dr. Dre, and listed here is 13 or so phenotypes related to vascular disease. And if we take a network-centric view of vascular disease, and obviously all of those markers of potentially protein and whatnot phenotypes in the blood that you can pick up, or even at the most basic level of genome, and you might be able to pick up. But for whatever reasons, I think to a large extent has to do with our common evolutionary experience, that a lot of those markers are in fact shared for multiple pathways, and our real challenge is identify some of those major and critical supernodes using Jay's word and drivers in these interrelated networks for disease phenotypes. Now, again, that led us to, again, step back. I wanted to, again, take a more broad view about this issue. And several years back, this is the typical analytical epidemiology that we have taken to summarize several decades of this epidemiology works and see if we can identify risk factors for whatever disease that you have all heard about with the metabolic basis for it. Here is summarizing all these common risk factors for type 2 diabetes, CHD, colorectal cancer. And if you look down the list, some of these major risk factors are fairly easily identifiable. I mean, it's be it because the way that measure is good, or because it's just the magnitude of the association is so big, it's actually quite easily identified. But the challenge started to show up when really, as Ross was alluded to in his talk, when we look at the complexity of our environmental exposure, many of which are also subtle and change over time, the challenge become. But by and large, you still, with more study devoted to the area, some general consistent patterns still can be identified. And all of which, again, help us. The idea is that that's still not satisfactory enough, but the consistency of which also suggests to us that to help us to break into the so-called black box is the fact that indeed there's some common big patterns that can have physiological implications, and one of which is illustrated in these figures that we have articulated about a decade ago, is this process of interaction between our genome and the environment that somehow increase our risk of insulin resistance. In other words, our have becoming an increasingly metabolic efficient population, and subsequent hyperglycemia and compensatory hyperinsulinemia can not only, from one hand, lead to what we all recognize now, glucose intolerance and type 2 diabetes, but also for other pathways such as dyslipidemia, hypertension, and other hemodynamic changes involving inflammation and thrombosis, and ultimately causes heart disease. Now, this kind of pathway and breaking into the black box also help us to think about in terms of the responsive to the environment, what are the potential critical, again, supernode, as well as major drivers in picking up those exposures. And also this is echoed to what Ross was discussing earlier in the morning. And because of the complexity, I think it would be good for us to take it back and to take advantage of whatever data is available. And this data is actually summarized from years of nutritional anthropological works. If you took a look at the entire evolutionary history of humans, you will see some major changes of dietary component over time, but none of which are more significant than the two revolutions. One is the agricultural revolution, and another one is the industrial revolution. And then those authors actually identify 7 major components of which in the human diet that have changed more recently, that presumably this kind of modern dietary exposures are really the main interfering forces colliding with our ancient genome that causes the disease of the modern cultures. So as a result, that from intervention or from a prevention perspective, one really ought to be looking for are potential dietary advantages that would be good for our ancient genome. And so they does give us some hints in terms of how we should go about looking at this. And as those works indicate, in one of the #1 changes, the dietary glycemic low. So we are one of the first to sort of like take advantage of those information and see how we can apply it to our works to better capture that kind of dietary exposures. Now, for example, if you put someone in a diet with pure chemical concoction, is glucose, adipoleptin, and aminopectin and aminos, which is basically some different type of starch, and you see this kind of fairly consistent change. And obviously, the postprandial glucose itself can immediately suggest to us be used as a biomarker that would allow us to capture that kind of aspects of diet. Okay, for example, refined carbohydrate is one of the major aspects. Now, trying to further illustrate this concept is that if you take the entire process of food production all the way to what the human response is after injection, you can argue that there's multiple of in vitro measurement that you can do to capture that kind of effects. In fact, Han Angus group in Cambridge University have done quite a bit of works in measuring the variations of different types of starch, even with the same total amount in terms of gram. And yet find out that just simply because of the structure of the chemical reorganization is different, that leads to different response of a glucose response in the human body. But then ultimately at the end, you can use a marker to capture that. And in this instance, it's simple glucose. It's almost like, whereas obviously there's many factors that would affect the actual food itself, or the characteristic of the food itself would actually affect the ultimate response. But ultimately, you can see this as almost like a bioassay done on each and every human being. Then you pick up that outcome. Okay, so it's one of which is proposed by David Jenkins' group at University of Toronto in the 80s to capture this kind of quality effect measured by glucose. Now, that said, there's also complexity involved in here. Immediately after taking a meal supposedly containing refined carbohydrates, obviously glucose is, postprandial glucose is the best markers, but long-term wise, that human body also have a remarkable ability to convert and metabolize those nutrients. And take here, in fact, that's what we found. This is data in the Nurses' Health Study, where we take a sample of about 200 healthy postmenopausal women who serve as a control in a case control study of breast cancers, and measure using a food frequency questionnaire of roughly about a year long of the dietary habit. And that's what we see about this kind of fasting, triglyceride effects associated with intakes of a high glycemic load diet modified by their BMI status. Okay, and this is actually quite consistently being validated by some of the metabolic trial when people were put on a high carbo refined carbohydrate diet versus the high fat type of diet. You see this kind of response. So the biomarker itself actually changed for the certain type of, for the same type of dietary intake. Now that leads us to follow up on this, ultimately using disease as an endpoint when we apply the concept of glycemic index into assessing the dietary load, glycemic load of the our own cohort participants, and that's what we've seen, this kind of phenomenon. In fact, you see the risk of heart disease over over a 10 years period actually roughly double, and especially among those who are already overweight to begin with, who presumably have a greater degree of insulin resistance, and as a result may not be able to tolerate that kind of diet. Now that's just one example of applying the biomarker to a better measurement of dietary refined carbohydrate. And I mentioned earlier another major important principle, a biological principle, is this interaction. In fact, there's a lot of the nutrients in the body's the homeostasis process is being controlled genetically, and this is one of the works, one of the papers that we published several years back now, when we show that, for example, in the body's absorptions of magnesium, and magnesium is also one of the minerals that's most consistently emerged from some of the observational works that we have conducted in the past 10 years looking at type 2 diabetes. See here is that obviously the magnesium intake does affect the absorption process, but not necessarily in a completely linear fashion. And in fact, it was really controlled by both passive and active transports. Especially the active transport process was controlled by two major genes called, which is ion channel transient receptor potential, with fairly big genes, one called trypsin 6 and other one called trypsin 7. And then it is really when the magnesium intake, the passive intake is not enough, when the active transport potential of the human being become more important in disease risk. So we took a look in our own sample to search, just like I mentioned, those genes are pretty big. Just within this gene of trypan6, we genotype about 20 or so the so-called haplotype tagging genotype for this gene, and then search for its potential mutations that might be related to type 2 diabetes risk, but we were not able to in the entire sample. But when we really separate the groups into different magnesium intake level, that's when we see some suggestion of significance, and really show here is the risk of type 2 diabetes roughly double among the groups that have low magnesium intake in the lowest quartile levels of magnesium intake in combination of a combination of two non-synonymous mutations defined here by the haplotype TC. Now, this is also, and then we subsequently trying to validate this with a small pilot trial where we feed folks with magnesium versus placebo, and we do real-time PCR in peripheral monocyte to see this increased expressions of both genes indeed. And now that's not even the whole picture, and obviously, if in the small samples that are looked at, that we clearly see that in response to magnesium or placebo, then there's a whole array of gene expression, gene being expressed differently between the two groups. Now, fast forward to move even further down to the genomic scheme. Here is, most of you know about the recent advance in the whole genome association arena, have identified for the genetic of type 2 diabetes as a whole set of genes, and specifically SNPs, that are directly predictive of diabetes risk. Here is already a bit outdated. Now it's somewhere around 18 or 20 or so loci that have been identified, but know that most of the magnitude of association, as I point out in that network slides that Tom gave me, is a lot of them are really modest, with the biggest TCF7L2 here with a relative risk of around 1.37. And this is data from the Framingham Heart study that published about a year ago in New England Journal of Medicine, when you see those researchers are very careful when they build a genotype score incorporating all this whole genome association identifiable markers and create a genotype score, which indeed can help predict one's risk individually in terms of getting at the calibration issue of about, I think here, about 15 or 20 years of cumulative risk of developing type 2 diabetes. But if you wanted to, based on the genotype, to discriminate the two populations of the diabetic from the non-diabetic, it is almost useless for this kind of markers out there. Now, so let me just very simply sort of at this point summarize this before I move on to the next stage of biomarkers, where we review it in the context of the disciplinary in a historical context. So I think it would be important to keep in mind, though, even when we talk about biomarkers, it does present sort of like the objective aura, but the same kind of, there's obviously some good characteristics of biomarkers. It would help to improve specificity as well as the sensitivity for measuring both exposure and outcomes, and it will also help us to identify individual susceptibility or individual risks of developing certain disease. In other words, to calibrate that individual risk better, while simultaneously reviewing some of the potential pathogenesis related to that particular disease. Those are all good, but certainly there's other bets that come along with it, one of which is the difficulty in obtaining, storing, assaying those samples. And in fact, it might actually introduce biases into the biases that we wanted to eliminate it. Okay? Now, that said, why do we still want to build this kind of predictive model? And that argument has been well articulated. Obviously, you need to find, sometimes it may not be that intuitive and straightforward when you have all these biomarkers available and you attempted to measure it, but the disease itself, the specific phenotype that you wanted to study, really also its own inherent characteristics are extremely important in the total equation when you evaluate whether or not it would be helpful to build a predictive model. And in the case of diabetes, I think it is actually fairly well articulated. Now, this is the work that we have been involved in the past few years now. In fact, it has not yet been published in the Women's Health Initiative, where we're trying to build a predictive model that would better discriminate as well as calibrate type 2 diabetes risk in the Women's Health Initiative. This is only for 6 years, where we incorporate all the traditional risk factors, family history, age, BMI, and some of the inflammatory as well as endothelial dysfunction markers. But you can see we already look pretty decent, including fasting glucose here, in terms of getting at somewhere around the C statistic over 90% for type 2 diabetes across different ethnic groups. Apparently, among white Caucasians, the discriminant abilities was the best. But even so, and that's the reason why we haven't published this yet, is I feel like this is somewhat of an overestimation. The way how we deal with the diabetes and is based on really glucose level. So once you put glucose into the model, it almost specifically defeats the purpose of prediction. So you use the criteria of glucose, either fasting or postprandial, as also a diagnostic criteria being used in the predictive model. And there's some problem in that if we truly wanted to get at the disease at an early stage, we need to find other markers. Now, that's another important consideration come into play, which is the clinical observation. What do we know that are the most salient clinical feature for the particular phenotype of your study in your biomarker of identification? And this is what leads us to think about sex hormone and sex hormone binding globulins as a potential diabetes, important diabetes predictor. That's something somewhat have been overlooked in the diabetes community. And that's what I wanted to talk a little bit about our recent studies, how we come about studying this. Now, if you took a look at, again, the literally 4 or 5 decades of the analytical epidemiology literature out there, you will see this kind of general patterns I mentioned earlier, that no one would deny that obesity is the single most important risk factor for type 2 diabetes. However, if you look at all this summarize this prospective data, you will say even though obesity is the single most important risk factor, but it does not affect men and women equally. It appeared to be stronger in women, as you see in this slide. That is essentially a meta-analysis, but stronger than men. And once the women develop diabetes, its risk of developing heart disease actually double that of men. So all of this is a suggestion, or at least an indication, that sex hormone, there might be some biological underpinning for this kind of phenomenon. Now, I should also point out that there's an entire different field of the so-called health disparities, and folks have been trying to study to find out exactly whether or not it's our access to healthcare that leads to this kind of phenomenon or this kind of sex disparities between men and women. But our argument is that there might be also biological component behind that, and that also is what lead us to go about evaluate the literature a little bit more carefully, while we identify a ton of the clinical observation that, especially in the PCOS literature, showing that folks with insulin resistant and also a high level of androgenicity tend to also have diabetes. But those are clinical observations, and we also identify fairly large heterogeneity in the literature. And unfortunately, also genetic data are very lacking, especially the kind of genetic data that some of you have seen earlier that we really help to illustrate whether or not some of the factors are potentially heritable and as such causal. Now, so then to our advantage, we again, to just echo what Ross was alluded to earlier, the importance of this kind of investment into doing this kind of prevention studies. And to our advantage, while I was at Harvard, I was involved in two a long-term intervention trial. One is the women's health study, another one is the so-called physician's health study, both of which were designed to evaluate the clinical efficacies of aspirin and some of the vitamins for long-term chronic disease preventions. But we also collected blood and DNA sample, which allow us to comprehensively examine this set of hypotheses that we wanted to. So when the data that come along that is suggestive or significant, then we wanted to follow up with some potential experiment to verify those clinical as well as epidemiology observations. And that's some of the data that we have reported in the past few years. Here is the extradiol data that show in roughly about 750 case control samples, and divided by the estradiol level in the blood, and you can see here by quartile, and already emerged, again, this is about 10 years long-term follow-up, incident cases of diabetes. In this women, we see a relative risk after adjusting for matching factors and all the traditional risk factors for type 2 diabetes, 5-fold increase in risk, which was quite interesting observation that had never been reported before. And then in the literature or the classical teachings in this sex hormone field is the actual free hormone that's important. So we also look at whether or not the free estradiol really is predictive, but slightly we see a stronger relative risk, but not by much difference. Now, when we look at estradiol in men, obviously it's capturing, I should say that this is also already adjusted for BMI or waist circumference in the data. Again, somewhere a LIFO increase in risk, which doesn't seem to have any differences in terms of whether or not it's total estradiol or free estradiol alone. Again, in men, we don't have as much data, so we, instead of categorize the data into quintile, we categorize them into quartile. Take a look at the testosterone level in women. It's a different story. It seems that the effects of testosterone in women increase its risk of developing type 2 diabetes. And SHBG, when we look at the free testosterone, you see that SHBG seems to play a fairly significant role in those women. Whereas in men, we see a completely different picture. Testosterone appeared to be protective, if anything. The relative risk is only 0.13. And yet, once you put in the context of SHBG, it would be even more important. So that's what leads us to take a look at the data in SHBG and comparing. Again, this is in women, the two extreme quintile. That's what we see, a relative risk close to 20. Okay, this is 20-fold risk difference. This is in men. We cannot categorize it into quintile. Instead, we categorize them into quartile, somewhere about 10-fold differences in risk of developing type 2 diabetes in 10 years. Now, looking at, again, trying to stratify and looking at interaction to see whether or not it really is as what we were told according to the textbook, that is the free hormone is at play here. We look at the interaction, and it appeared that, again, the interaction is not significant, and much of the effect really come from SHBG. This is in women, but the same in men. Now, that's what leads us to then follow up if an effect that's that strong so later in life, and those are mainly postmenopausal women as well as age comparable men, all of whom are apparently healthy at baseline when we started the study. So that actually suggests to us that it's important indeed to look into the genetics. So we identify the genes, and this is some of the genes actually of SHBGs are fairly well described and sequenced already, primarily of the works done in the cancer field. So we then pick based on the data that have been sequenced. Those are sequencing data, and we identify this set of alleles to genotype in our own data. Again, like I said, the gene is actually not that big. It's only got 3.2 kbs. Then what we find is these papers that just published this past month in New England Journal of Medicine very clearly show that the two alleles that we identify, one is the RS6259, another one is RS6257. One is in exon 8, another one is in the promoter regions of exon 2, both of which seem to have functional, and obviously this has functional significance in terms of actual chain coding of the protein itself. And for this one, it seems to be affecting the alternative splicing. And most important of all is that their effects on the circulating level of SHBG seem to be additive. One is increasing about 10 or 12%, whereas the other one is reducing it by the opposite 10%. So both of them get together is somewhere around 20% elevations in the circulating level of SHBG. This is, again, genotype combinations, and what that translates into is the relative risk of somewhere around 0.43, somewhere around 57% reduction in risk associated with this combination genotypes alone. Now, that is another way to because Ross is here, so I have to show some causal modelings to impress him. So this is a somewhat oversimplification of the causal model we look at. Then most of the biomarkers that you heard about is in the circulating level that you can identify a lot. Then the parameter of interest essentially is here. One we wanted to capture is this beta-1 for type 2 diabetes. However, as we all know that for plasma phenotype in response to diet or gene and whatever environment, it would obviously change, and some of which we measure, the transitional risk factor for diabetes, some of which we don't know. They nevertheless all would affect this. However, if we have information of alpha-1 here where we directly identify a genotype that walks through these pathways, because genotype is determined, especially those are germline mutations, determined at birth, okay? That presumably cannot be affected by those confounding factors. So now that's obviously given certain caveats here. I listed here the genotype must have robustly associated with the particular phenotype here in SHBG level, and it really has to affect the disease through the particular phenotypes. Then with those assumptions, then we can apply to do this. Another way to put this is another, in my view, the application of the biomarker at this genetic level to help the study design to some extent. Again, I hypothesize, this is totally make-up hypothetical examples. The randomized trial, supposedly we wanted to give participants with competent SHBG to treat them, to prevent for diabetes, okay? But through the randomization process, you wanted to make the unknown confounding factors evenly distributed between the two groups. So one group would accept the treatment, another group would be placebo, and ultimately you measure the effects that would be independent of whatever potential confoundings. So the same concept here is in the Mendelian randomization process because of, again, the Mendelian law of random assortments determine that given those assumptions, if those assumptions fulfill whatever the unknown confounding factor that are socioeconomical should be evenly distributed based on those genotypes, okay? So that then be, if we incorporate that in our modelings, then that would potentially help us get rid of those confounding factors. That's exactly what we did, and using this newer form of analytical techniques where we identify for SHBG still somewhere around 0.39 of relative risk for type 2 diabetes, okay? Now, ultimately, we wanted to incorporate everything, incorporate the traditional, the genetic and biochemical predictors for type 2 diabetes to build the kind of statistical models like this to see whether or not we can improve predictions, because diabetes is a very much preventable disease, even at a late stage of glucose or some impaired glucose intolerance. And then the other model is trying to synthesize. Ultimately, we wanted to test it still, whether or not when we articulate this kind of hypothetical models of androgenicity, simply defined with one single markers, because we believe this single marker of HBG can not only serve as a marker for susceptibility at the genetic level, but also can be manipulated at its plasma phenotypic level and ultimately can be responsive for disease manipulation. So we wanted to test this and to see whether or not modulation of this targeting SHBG can indeed affect not only impairment in insulin secretion, but also insulin action as well. And this is some of the works that Kathy Lee have done. This is some preliminary works, but with 34 subjects that enrolled through our GCRC, fairly interesting association that have been shown for now, and we do know that given estrogen itself can significantly increase hepatic production of SHBG. I mentioned earlier about the glycemic load, the monounsaturated fat, monosaccharide as well, as well as some of the high fructose corn syrups that we have been taking in increasing our total glycemic load. And then I now also describe to you some of the classical medical textbook teaching of SHBG as just a bystander carrier that very much influenced by insulin or obesity. And this is some of the works that have been done by a group in Canada. We recently published in JCI, which helped to show very interesting different mechanistic interpretation of the data. Shown here, it's very clearly in the HEPG2 cells lines when they injected simply glucose and fructose directly, you see this clear suppression of SHBG. And what's even more interesting is that the mRNA level, I think here is just a simple real-time PCR was being done, showed that, again, the SHBG was suppressed, which is quite interestingly directly by adding those monosaccharides, and also one transcription factor that being also suppressed, which is called the hepatocyte nuclear factor 4 alpha, which also has been shown to a form of type 1.5 or type 2 diabetes called maturity onset diabetes of the young, that has direct effects on impairments in insulin secretion. what they go on further to show that, in fact, insulin does not even seem to affect the human SHBG production whatsoever in cell line, as well as some of the Western blot analysis they did. And again, most importantly, confirmed that it really is this transcription factor seem to activate the promoter of the SHBG in hepatocytes expressions. Now, this is interesting. What I have trying to use the next 5 minutes or so to help put everything together, and why I think it's so important that we need to bring all the disciplinary together. And obviously, the field has been extremely, there's a lot of advances in our field. And it is very clear that both, be it in public health or in medicines, and you really have to understanding the molecular aspects of the biology. How can we sort of like integrate this together would be both a challenge as well as opportunities. We are truly to be able to answer some of those questions, or age-old questions of how and why, and how can we actually get to the bottom of it. And I don't pretend to have a solution, and at least this newly formed inter-school program is one of our way to sort of meet that challenge. Use here is one example. I wanted to lay out some of our sort of different paradigms of thinking between the quantitative population science and some of the mechanistic biological science here. I used this cartoon show of a beta cell, something that had actually been worked out for several major family inheritance of maturity onset diabetes of the young. This kind of, in a handful of the family that have autosomal dominant mutations, that biochemically each one of the single gene mutations that then be mapped back to the specific pathways or specific pathways that can illustrate the immediate biochemical understanding of the disease itself. Now, some of this classical human genetic and biochemical works is sort of like illustrated beautifully back in the 70s it's, already. But even at this level of interaction, some of those are really interaction we are talking about, the gene and gene interactions, and they are low here, there's glucose sensing and transporters in the beta cell itself. And how do we then translate, even for the genetic interaction, into a population level when we see this kind of patterns? Some of which, once we introduce the measurement error, as Ross pointed out in his talk, of those complexities of diet, that would even become more challenging. And it is clear that we should not forget the size of prevention. And there are different strategies to do that. And I listed here a formula that most of my students in public health, in epidemiology, are very familiar. This is essentially the so-called population attributable risk on the left-hand side of this equation, which really is a function of the prevalence of the particular exposure of interest plus the etiological factors, the odds ratio, the rate ratio, the relative risk, that I would argue that the biomedical community pay a lot of focus on this particular RR itself, the particular etiology agent for the disease, and yet do not pay enough attention on the PAR, okay? But nevertheless, really is one cannot really sustain itself without the others. And that's how I see this. It's important to keep in mind that what is the ultimate benchmark upon which we will use to evaluate our work and allocate our resources in doing this work of the future. And this then leads to this slide of my own imaginary model of resources allocation. And that really have to, again, to be addressed to just echo what Ross was saying in his keynote talks, that why, in our nation at least, that's not as it is. Huge amount of resources, not only in healthcare, but even in biomedical research, is devoted into doing this population. Whereas the quality of the evidence were mandated by law that it's important. That's the reason why some of the pharmaceutical companies telling you that they take $850 million to devote one particular drugs, because they are required to do the kind of double-blind placebo-control trial to evaluate phase 1, phase 2, phase 3, phase 4, very carefully mandated by law to do that kind of works. And no wonder a huge amount of resources are devoted in this area. Whereas the kind of population, especially prevention trial, as the type that have been led by Ross, such as the Women's Health Initiative, are not the focus of support by allocating appropriate resources to it. And nevertheless, those are extremely important, for it affect millions of people, millions of people. Okay, so I wanted to end with this, my own 3 bullets of the ultimate benchmark. First, prevention of metabolic disease, I think, has a huge nutrition component, is of utmost importance, because it's the iceberg, above which the tip are cardiovascular disease, some cancer, diabetes, all are tip of the icebergs, and we need to attack that iceberg underneath. Ultimately, that's the only way to curb the epidemic. However, we should not see the biomedical enterprise as somewhat competing, but rather complementary. In the process of addressing this epidemic, it is extremely important to keep in mind that prevention does require the best possible evidence to define not only the strategy for prevention, but also understanding the biological mechanisms. And within that context, we should also keep in mind we don't hide up what we have not yet delivered. So with that, I wanted to thank my group here at UCLA for the past 4 years since I moved my base here from Boston, that I have gathered a group to help, without whom this work is not possible, and also some of my colleagues still at Harvard that has continued to support and continue to collaborate, and fundings from various institutions. Thank you very much.
Thank you, Simon. It's very interesting about the role of SHBG, and I need to compliment you for that. I was also wondering whether there may be other potential explanations you may consider. One of the things I think also is happening, at least one of my postdocs in the lab has been doing, is just the withdrawal of estrogen without any high-fat diet, just a normal diet in black 6, leads to things like the metabolic syndrome. They develop glucose intolerance, insulin intolerance, increase in visceral fat. Now, what is interesting is that when, at least in humans, when one tries to completely correlate with SHBG, one has to consider other facts. For example, in men, it is the orchidectomized men that develop metabolic syndrome and not the test is intact. And also Simpson has shown that in male mice who are aromatase deficient, they develop metabolic syndrome. So it looks like it's the conversion to estrogen which is so critical. And some of the studies we've done with Jake on atherosclerosis and others tend to indicate that withdrawal of estrogen, just the withdrawal of estrogen, no different diet, can lead to an inflammatory-like reaction which could lead to these effects. Because what happens is in rodents, there's no SHBG, there's androgen-binding protein, and yet you see with the withdrawal. So SHBG maybe, is probably important, But along with that is the levels of the female hormone, which may also play a part.
Well, I totally agree with you that it is estrogen obviously is important, but because of the pathway is so complex and our understanding is so incomplete that you never know until you do a formal trial, especially it's really hard to understand whether or not the model that you presented, in fact, a lot in the literature, there are many of these kind of different animal models, results of which, whether or not they can be translated into the biology of an intact human, is very challenging. So the goal ultimately has to be reevaluated in a clinical trial setting to understand whether or not, and that's essentially what the women's one major accomplishment of the Women's Health Initiative, for the example, in evaluating the clinical efficacies of HRT for vascular disease prevention among postmenopausal women. Now, to directly get at the question of whether or not SHBG really is causal, again, I agree, it's still a subject for debate. What I have presented is that some very powerful genetic evidence in support of that some of the Gem9 mutation we identify seem to be predictive of diabetes risk long term. And those Gem9 mutation obviously are determined at birth and won't change and would not be affected by the subsequent environmental risk factors. And then the incorporation of the so-called Mendelian randomization analysis further confirm our view on that. And that ultimately need to, I think because of this kind of consistent prospective data, we didn't see this in one cohort, we actually replicated in another cohort. And now, since our paper has been published, there's another group called the Diagram Consortium, which have studied 20,000 diabetics with 20,000 controls, where they basically replicate with the same identical analytical strategies of our findings. Now that speaks directly to the potential causal pathways that could be dependent on the pathway that you mentioned, or even just dependent on insulin, for example. But also, I would argue that potentially some as yet unrecognized pathways related to SHBG is totally possible.
I agree with that. SHBG is important from your data and others, but I'm saying not to forget the estrogen and the conversion of testosterone to estrogen, because both are linked. And one, for example, estrogen may, we know, increases SHBG, and that may be the way it's acting as well.
Yes, thank you.
Thanks.